showed that they could use expansion to start tracing circuitry of the brain, which is very exciting, right? One of my dreams is if you could make a very detailed map of the brain, you could maybe simulate it in a computer. And that's one of the things I'd love to be able to do, because that would allow us to maybe fulfill this question of like, what is a thought or a feeling? Because we could watch it, right, in action, and then in silico tease apart how it emerges from the fundamental building blocks.
Hello, everyone, and welcome back to the Neuroscience and Beyond podcast. I'm your host, Villain, and together with my amazing team of graduate students, we're on a mission to bring the wonders of science, especially neuroscience, closer to you. Today, we're thrilled to welcome one of the most pioneering figures in modern neuroscience at Boyden. As a neuroscientist, inventor, and entrepreneur, Professor Boyden is known for pushing the limits of brain research. He holds the prestigious title of Y. Evatant Professor in Neurotechnology at MIT McGovern Institute and is also an investigator at the Howard Hughes Medical Institute. His lab focuses on developing groundbreaking tools to analyze, repair, and simulate brain functions. Professor Boyden has received numerous prestigious honors, including the Breakthrough Prize in Life Sciences and the Brain Prize. His work on optogenetics and expansion microscopy, two technologies considered Nobel Prize worthy, has revolutionized the field. In this episode, we discuss his key innovations: optogenetics, expansion microscopy, and multiplex imaging. Multiplex imaging in particular enables researchers to track signals from living cells. We will also explore how these tools are being shared with the world, opening new paths toward transformative therapies addressing brain disorders. Before we begin, I'd like to mention that this podcast is a separate activity from my position at the University Medical Center Göttingen in Germany. This podcast is supported by the International Max Planck Research School for Neurosciences, the Cluster of Excellence Multiscale Bioimaging, and the European Neuroscience Institute in Göttingen. If you'd like to support us, please subscribe to our channel and do not hesitate to drop us a comment under our videos. And now, dear listeners, please enjoy the conversation with Professor Ed Boyden. Professor Boyden,
hi,
it's a huge honor to have you here. I'm good, thank you. How are you?
Doing all right, yeah.
You had a long flight from Boston to Göttingen. I hope you're not jet-lagged today.
Still a little bit, yeah.
So a little bit. I'm very excited and looking forward to our discussion.
Me too.
This time, I'd like to open with a little bit of statistics. So you published your first work in 1999, and it was about physics, quantum computing. Since then, you've published more than 160 papers. You have nearly 300 patents. And I want to ask you, when do you sleep?
Well, sleep is pretty important, I think, actually. I like to load up lots of questions in my head, and then sleep, I think, actually helps process them and generate creative solutions. So when I wake up, sometimes the solution has popped into my mind. And so I think sleep is a great tool to help with ideation and the maturation of thinking.
I saw you just taking a note. You had some idea, you typed it in on your phone. Do you have any notebook next to your bed where you write your notes when you wake up? For example, your ideas that you had during dreaming or before that?
I guess I use my phone these days. In the past, I would write with pen and paper and then photograph the pictures. And I probably have tens of thousands of those pages. But typing on the phone makes it more searchable.
Yeah, that's incredible. So let me start with a little story about how your work influenced my life and my present life that I'm actually in Göttingen. So in 2003, around that time, you worked on a very powerful and influential technology called optogenetics. And you published your work, and since then, this field basically exploded. Fast forward to 2020, when I had my interview for this grad school in Göttingen, the IMPRS for Neurosciences. And one of the very important questions that I was asked is, what is optogenetics? So luckily I was prepared, and I think I gave a good answer, so therefore I'm here.
Great.
Now I have the pleasure to ask you, what is optogenetics?
Well, brain cells use electrical pulses as part of their computational machinery. And if you could stimulate electrical pulses, then you could test causality. You could ask whether certain patterns of brain activity are important for behavior or for a disease for that matter. Now, electrical stimulation is very powerful. It's been used to treat people with Parkinson's disease, for example. It's been used in basic neuroscience for over a century. But electricity goes in all directions. And with optogenetics, we can aim light at individual cells, or just one kind of cell. And you have to make the cells turn light into electricity, the equivalent of little solar panels that you would install on the cells. And we adapt them from the natural world. So all over the tree of life, you can find molecules, proteins that convert light into electrical signals. We can put the gene into the brain cell using any one of a number of standard gene delivery methods, gene therapy methods. The neurons will then manufacture the light-activated protein, shine light, and you can get light to electricity. Electricity stimulates the brain. So it's called optogenetics, opto for light, and genetics because these are genetically encoded molecules.
The field has developed, of course, enormously. And at the beginning, you were able to activate neurons by shining a certain wavelength. And now you can also inhibit the activity. Is that right?
That's right, yeah. So in 2007, Xue Han and I published a paper together on using light-activated chloride pumps to silence neurons. And then in the years since, we and a number of colleagues and collaborators have been trying to make the molecules as strong, as fast, and as minimally invasive in terms of the instrumentation as possible.
So you summarized very nicely what optogenetic is. Can we maybe step back a little bit and talk about how the idea was born, who worked on this, and also the history behind the channels that were discovered?
Sure. Yeah, so I started graduate school at Stanford, 1999. I trained as a physicist and engineer beforehand, and also studied chemistry for a couple of years. So I had a very long undergrad period, 6 years. So I got to learn lots of different fields. And I started graduate school working with Dick Chen and Jennifer Raymond at Stanford, and really was excited about tool development. Could we build technology that would let us map, control, look at the brain? And so my very first year of grad school, I met a fellow student, Carl Dijsselroth, who was finishing his MD at the time. And we hit it off and started talking about what could be done to stimulate the brain. At the time, he was doing transcranial magnetic stimulation. And we decided to try to see if we can make it very precise. And so we used a method that I still use to this day. We try to think of every way of solving a problem. So there's only so many kinds of energy you can deliver to the brain, right? There's sound, there's light. That's about it. So light comes in different flavors, radio waves all the way to X-rays. Sound could be mechanical force or ultrasound or things in between. Anyway, you can write down the list of every way of delivering energy to the brain on a single sheet of paper. And so light had a lot of appeal to me because, it's, of course, as fast as anything ever gets, and you can aim it very precisely. And then what clinched it was I was reading these papers about microbial opsins or rhodopsins. So unlike the molecules in our eyes that sense light, these are 7 transmembrane proteins that convert light directly into ionic flux. And single-celled microbes use these to convert solar energy into chemical energy. And what clinched it for me was a paper from a group in Japan that showed that these molecules, which at the time were only found in halophilic species, so microbes that live in very salty water, they found one molecule that, for whatever reason, worked at low salt concentrations, the kind you'd find in the mammalian or the human brain. And so I started emailing people in spring of 2000 to start collecting these reagents from people. But it took us a while to actually get around to doing the experiments. So I think around 2003, we started really thinking about doing the experiments in earnest. And by summer 2004, we had the first data point. We put one of the opsins, channelrhodopsin 2 from algae, which was discovered by Peter Hegemann and Gerd Nagel and Ernst Bomberg and colleagues. We put that gene into a cultured rodent brain cell in a dish. And if we shine blue light on it, it basically worked on the first try. We could get it to fire light-gated action potentials. And so that became the basis of our first paper that came out in 2005.
That's incredible. How did people react to your idea to start working on this kind of channels that are expressed in some other animal, and then you want to manipulate the brain? Was there like any encouragement? Oh, that's cool, or like it was more, what are you doing actually? Why are you losing your time here?
Well, in the beginning, I think a lot of people were skeptical of neuroengineering. At the time, a lot of neural tools were known for being prototypes, but a fairly small number of them were widely used, right? So patch clamp, of course, two-photon microscopy and GFP, both were developed around 1992 or discovered around 1992. Of course, MRI and functional MRI were characterized around the 1990s as well for the brain. But the idea of a discipline around neurotechnology was, I think, kind of a foreign concept at the time.
Mm-hmm.
So yeah, people were a bit skeptical. A lot of places I applied to for faculty rejected my application, and that's how I ended up being a professor in part of MIT School of Architecture, an interdisciplinary department called the Media Lab, where I spent many years. But yeah, it was a bit of an uphill battle in the beginning. I think a lot of tools were seen as not really solving problems. And so when I started the group, I really wanted to make sure that we were thinking backwards from problems, but trying to connect those problems to the ground truth, the underlying building blocks and how they work together. Mm-hmm. And those are the extremes, right? The problems are at one end, and the ground truth are like the minuscule, even nanoscale building blocks. And how do you connect that vast gulf? And that's really what our group set out to do around 2006.
What kept you push forward despite this kind of lack of recognition at that time? Was there anything like the strong belief I will do that, or was there anything else that helped you go through this kind of, let's say, hard period?
Well, I think I was obsessed ever since I was a child with the idea that we needed to understand human existence. And these philosophical questions, of course, have been tackled for thousands of years, right, through different approaches. But I got more and more intrigued by the question of whether you could tackle them through science. Could you understand the basis of things like the human mind or the human brain or physical existence, and then tease that apart using known scientific principles? If you can explain these complexities with known scientific principles, that's great. And if you can't, if you find something that is completely unexplainable and you can prove it, that's also nice. Mm-hmm. So you mentioned quantum computing earlier. So I was always drawn to projects that were at the border of philosophy and science. Quantum computing also is in between like mysterious things like superposition and entanglement, but also practicality like computing. And so my research experiences, which started pretty young, I left home when I was only 14 and went to a college or university in the state of Texas, which allows you to skip a few grades and go right to college. Mm-hmm. So I worked in an origins of life group. They were trying to create DNA in inorganic material like clay. And again, at the border of philosophy and science, if you will. But yeah, I got hooked on the idea that you could try to address philosophical questions through scientific means from an early age. And origins of life and quantum computing were, I think, too difficult. We still don't really understand where life came from, and we still don't have very good quantum computers. But yeah, I guess the third try was a good one. And it turned out the physics and chemistry training that I had from the origins of life and quantum computing worlds was just what I wanted to bring to bear on neuroscience. So it felt like a good path if I could, find the strategy to bring it off. And yeah, there were lots of difficulties in the beginning, but that was good training, I think, for understanding how do you galvanize a field, how do you align incentives, right? So rather than persuading people of the idea, why don't we just share all the tools freely? And now we've shared tools, I don't know, tens of thousands of times. And once people adopt the tools, then, which they would do out of their own self-interest, all the incentives are aligned, right? No need to persuade people. People use the tools because they want to do good science. And then eventually then people will see how the field has changed.
That's incredible. So a strong inner drive to understand life. And you went from the hard problems of quantum computing and origin of life to the easy one, stimulating the brain electrically. I think now it's a good point to ask you about the real applications of optogenetics. You mentioned that you can stimulate brain cells, but what can we do with that?
Well, stimulating the brain allows you to address causality, and there's two general reasons why you'd want to do that. One is to prove that something is important. So you can look at the brain all day and see electrical pulses, but how do you know what's important? Maybe an electrical pulse is just a copy, and the real action's happening elsewhere. By stimulating, you can test whether driving that activity pattern is sufficient for a behavior or a pathological state, or you can delete it through neurosilencing and see whether that pattern is necessary for a behavior or a pathological state, or the treatment of a pathological state. So causality is very much at the core of why optogenetics is interesting. And of course, a second case would be therapeutic ideas. So if you do have a pattern of activity that you can drive and that could help with a pathological state, then that could potentially be a prototype of a therapy. You could try to copy that pattern through non-invasive means, like sensory stimulation or electrical stimulation. Or just a few summers ago, a team in Europe also has been trying to put optogenetics into the human eye for treating blindness. And so, of course, being a gene therapy, using it in the human body is very challenging. And so a lot of the translational impact of optogenetics is in the study of the brain, and then to see if you can take that insight and implement it through a non-invasive or less invasive means. But it's interesting to see a second and maybe a little bit less expected impact, which would be the use of optogenetics in people to directly try to treat conditions.
Actually, this second aspect is something that we can talk about a little bit later because it's incredibly interesting. But I would like to continue again on the path of electrical stimulation of the brain of the cell. So let's say you stimulate a certain cell, which will kind of perturb or not a certain network in the brain, and maybe a system in the brain, the whole system. Can we say that by stimulating 1-2 cells, we can manipulate behavior in mice, for example?
Yeah, so probably at this point, thousands of studies have been done where people will activate or silence a set of cells, or sometimes even one cell, and you can see changes in behavior. So you can study sensation. You can drive one or more cells and see if an animal will respond to it. Can you simulate touch or visual stimuli or auditory stimuli? And lots of groups have done that. You could try to drive a set of cells or silence them and see if that creates or sustains or impoverishes or halts an emotion or the formation of a memory or the recall of a memory. And of course, you can try to drive or halt actions as well, right? Movements. And those can be done in healthy states or also disease model states. So most optogenetics is done in animals, such as mice. And you could try, for example, to halt a Parkinsonian symptom in a mouse model of Parkinson's disease, or like my collaborator Li Wei Sai at MIT is trying to drive patterns of activity to clean up Alzheimer's disease and so on and so forth.
Oh, this theta burst and wave activity of the brain. I think she works on this and in relation to Alzheimer, which is also another incredible topic. But
she's been doing 40 hertz stimulation, so in the gamma range.
Exactly.
You can
Sorry, not
cause,
theta, gamma, exactly.
And you can cause all sorts of interesting effects.
Yeah, that is true. She was also here recently in Neuroscience giving a talk, by the way, a couple of months ago. I should mention and shout out to the people from Horizons organizing team who actually invited Professor Boyden to come to Göttingen. And because of this, we had the opportunity to also invite him to our podcast. And we are, of course, very happy to have you here. Going back to optogenetics, let's say that at some point this technology will develop and we can deliver it to humans. We just mentioned that you can manipulate already behavior in mice. Do you think that we can manipulate also behavior in humans at some point in our future life?
Well, so a group has been trying to help people with blindness. So millions of people have lost the photoreceptors in their eyes. But the rest of the eye, of course, is still there. So you can take these light-activated proteins, put the gene into spared cells of the eye, and basically convert them into a camera. And so I think at this point, maybe a couple dozen people in multiple clinical trials are now using molecules from our group to try to restore vision. Retinitis pigmentosa, for example, is one condition that people are exploring. Now, what people don't know, of course, is this is not an approved therapy by any regulatory agency. And so pivotal trials that show safety and efficacy, are still ongoing. So potential concerns, of course, these molecules are from microbes and other organisms. Will the human body accept them, or will the immune system, fight back or whatnot? The eye has a degree of immune privilege, which is maybe one reason why the eye has been a first spot to look at. So to go into the brain, one would need a couple things, right? First of all, you have to have a hypothesis. Where in the brain would you stimulate? And I think that's currently an open question, right? If you study the mouse and you find a site that you can activate to treat a condition in a mouse model, how do you map that onto the site in the human brain? And that's an open question, I think. That has not been answered in the general case through lots of insight and through good old-fashioned scientific investigation. You can try to bridge that gap, but it's very much a difficult thing to do. Mm-hmm. Also, long-term questions about safety in the human brain, those have not even really begun to be investigated. So there's a lot of hurdles to be overcome, I think. But in principle, hundreds of thousands of people have electrodes implanted in their nervous systems. So I think over 100,000 people have deep brain stimulators for Parkinson's disease and tremor and other conditions. And increasingly, more and more kinds of conditions. So there are ongoing studies looking at depression, looking at obsessive compulsive disorder, all sorts of brain conditions out there. I think if there is a way to remedy a condition through neural stimulation, if there's a way to do it that's motivated well by a hypothesis of where to stimulate and what pattern to stimulate, then maybe it would justify the thinking about such a trial. But because there's a lot of unknowns, I think a lot of people have been trying to think about how to translate optogenetic thinking into, let's say, informed use of an older technique, like where to put an electrode or how to non-invasively stimulate the brain.
Mm-hmm. Just to make a short clarification, when you say the gene is expressed, we need to deliver this kind of genetic material, right? And the technique that is used nowadays is adeno-associated viruses, which kind of serve as a carrier, and the genetic material is packaged to bring this viruses, so to say, to the brain in this case, or to the retina, so that certain channels can be expressed, this kind of light sensitive channels, and then the brain should be stimulated by light. One of these, as you said, the retinitis pigmentosa experiments is done in the retina. We have in Göttingen a very interesting research group from Professor Tobias Moser, who tries to kind of bring optogenetics into the cochlea, and this is another interesting part. What do you think about that invention, for example?
Yeah, it's exciting. Yeah. We've talked over the years about what they're doing, and I think they're using some molecules that are based upon molecules that our group discovered, which is really exciting that we can help out. From their studies, it sounds like they can have more naturalistic hearing in animal models. So it could be quite exciting potentially.
That's incredible. Yeah, the idea there is that you, by stimulating your cells in the auditory pathway, let's say in the cochlea with the cochlear implants, the stimulation is not that precise. So the sounds that you're hearing is not the sound that we hear as people without hearing impairment. But when you have this kind of optical cochlear implants and optogenetics, you can precisely stimulate certain cells that can relay the signals into your auditory cortex, and probably or highly possibly patients with this kind of optical cochlear implants in the future, they might be hearing even very close to what people without hearing impairment hear, which is just incredible to think about it. If we continue in this notion, so you mentioned the deep brain stimulation and these chips, and I remember watching one of your TED Talks that was released roughly 13 years ago about optogenetics. Towards the end, you mentioned already back then that we would like to take information from the brain and put information back into the brain, and you talked about brain augmentation. I'd like to hear your take on the company created from Elon Musk called Neuralink, where they use this kind of advanced chips in the brain. And do you work on chips that are actually stimulating the brain with your technology, optogenetics? And is this even possible?
Let's see. So the company you mentioned, Neuralink, is putting electrically conducting wires in the brain. So they're using kind of an older kind of strategy, although it's been widely used in people. So, and then as far as I can tell, they're putting in arrays of wires to try to able to interface to more neurons. One reason why I think the optical method is exciting is that potentially you could avoid the need to put brain displacing objects inside the brain, right? You could imagine a projector and aim light, maybe through holographic patterning, and then you could stimulate cells. And then recently we also showed in mice that we could take modified optogenetic proteins. So Adam Cohen's group at Harvard had found that some optogenetic molecules act like weakly fluorescent voltage indicators. And so his group and now our group are also trying to improve their brightness. But anyway, if optogenetics can be used in people to activate neurons, you could also imagine using modified optogenetic tools to read out information. And because the molecules are very similar in structure. Mm-hmm. So yeah, I think the idea of all optical reading and writing from and to the brain is very appealing because potentially you could do it without having to displace any brain tissue. I don't think anybody has really solved the very long-term recording problem. Electrodes tend to build up scar tissue or gliosis and have other issues over very long periods of time. So that's an ongoing area of research. But one possibility is if you could just avoid putting things into the brain and park your device outside, that could be quite powerful. So our group isn't currently working too much on the hardware side. It seems like if other people can do something, then let's leave that to the highly trained professionals that can do it best. I think we're often trying to do the going from 0 to 1 and to get the first version of something going. But very often, a company or a group that's more specialized in a certain part of engineering can do a better job than us at a very specific area, especially if it's in an advanced part of a more traditional engineering field. And so chip design, I think, is an example where industry can do that far better than, let's say, a group like ours in academia can.
Mm-hmm. Chip design, and also probably I would imagine if you want to record constantly information from the brain, there is always the question about storage. So where to store this data, how to transfer it from the chip or the device. So this is probably another part that needs to be investigated. I remember here a group from Alexander Geil in the German Primate Center where they have very interesting setup. They have a lab with 29 cameras and monkeys with electrodes implanted in it, and they correlate basically the behavior from the camera, from the recording, with the brain activity. And always the questions that they discuss during the lab meeting is how much storage do we have? We're talking about terabytes of data within one experiment or something, and then you have to make sense of all of this data, right? This is probably where the AI will kick in. Speaking of AI, do you use some artificial intelligence tool in your research?
Oh yeah, I think of machine learning and AI as just like water or electricity. I mean, everybody can use it, and it's very straightforward to apply to many problems. And then certain problems, of course, require more advanced machine learning and AI. And so we're actually working to try to improve the tools as well for our purposes.
So you're working on many, many exciting things, and our time is limited. And I'd like to switch at that point the topic from optogenetics, although we can go back at some point, and go into another very, very exciting field called expansion microscopy. What is expansion microscopy?
So nanoimaging is very important. The building blocks of life, like genes or biomolecules or gene products, are all nanoscale things. And nanoimaging is a very important field in biology. So super resolution microscopy, cryo-electron microscopy have enabled lots of advances by enabling the imaging of very small things, such as proteins and so forth. But all these methods face two challenges. One is that they're difficult to apply to large 3D objects like brains or even brain cells, for that matter. And also, they tend to be expensive and require a lot of skill. So our thought was we often apply sort of what I like to call learnable and teachable creativity strategies. And one of my favorite is, can we do the opposite of what everybody's doing? And so, for literally 300 years, the way that you image in biology is some kind of lensing effect, right? You magnify some kind of information or energy from an object, and then that lensing effect allows the eye or the camera or whatever to capture that magnified information. And so we thought, what if we magnify the object instead? The first versions of the idea started with brainstorming with actually the very first postdoc who joined my group, Brian Charo. We were wondering, could you separate molecules from each other to decrowd them, to get better labeling? And so we started thinking about whether you could use things like hydrogels to expand objects. And then that was around 2007 or so. Several years later, two amazing then graduate students in the group, Fei Chen and Paul Tilburg, were in the group. And Fei was trying to do super resolution microscopy, and Paul was exploring electron microscopy. And that's when we really realized how hard nanoimaging was. And so we decided to go after expansion, and we started reading about soluble polymers and thinking about how to do the physical magnification, and we were able to make it work. So the first paper in 2015, we could physically magnify brain specimens about 100 times in volume, 4.5 times in each direction. And since then it's been a pretty constant stream of improvements as well as applications.
What is the most striking discovery that you have done using this expansion microscopy so far?
That we've done in our group?
Yes.
Well, our group mostly works on the tool invention, and then others tend to be the people who apply it. But it's been fun to see lots of people solve mysteries within their field. There are lots of examples where, for example, ribosomes, right? They make proteins in cells. For decades, they've been studied in dendrites in neurons. Aaron Schumann at the Max Planck in Germany showed that using expansion, they could find ribosomes on the other side, the presynaptic and axonal side, which was not, as far as I know, visible with electron microscopy. So that solved, I think that was a mystery for like half a century or something. People are using expansion to try to locate where viruses are hiding inside cells or how they move between cells. People are using it to make brain maps, so by expanding and labeling the wiring of the brain, could you make a map of the brain, so-called connectomics? So recently, several teams have showed that they could use expansion to start tracing circuitry of the brain, which is very exciting, right? One of my dreams is if you could make a very detailed map of the brain, you could maybe simulate it in a computer. And that's one of the things I'd love to be able to do, because that would allow us to maybe fulfill this question of like, what is a thought or a feeling? Because we could watch it, right, in action, and then in silico tease apart how it emerges from the fundamental building blocks. Early cancer detection, we published a paper in 2017. Early in a cancer, right, it's hard to do the diagnosis because the changes are small. What if we just make them look bigger? And so early in breast cancer, for example, doctors can disagree half the time about the diagnosis by expanding those biopsies, actual biopsies from human patients. We had doctors look at those and train a machine learning algorithm to classify them, and it was able to do much better than on the unexpanded samples. The list goes on and on. I think it's been used in over 700 experimental papers and preprints, not like review papers, actual experimental papers. So it's spreading very quickly, maybe faster than optogenetics, actually.
That's incredible. And the funny thing is that you develop a technology and you don't know what will be exactly the outcome. There are so many surprises, the cancer, the diagnostics, etc. And it's incredible to see that your work is actually used by thousands, as you mentioned, around the globe to solve not only fundamental problems in biology, but also in translational science and diagnostics and etc. And this is, I think, only the beginning, right? I'd like to pick up your brain on the connectomics, but I will come back on that and ask you about the technique itself. So when you say expansion, we know that in the brain, the cells are very crowded. You have neurons, you have glia, and you have also blood vessels, and everything is very, very tight together. And by expanding it, you basically make things that are invisible to our eye visible, so to say. But how do you preserve the tissue from rupturing, for example? And how do you know that, okay, when this was actually intact in the brain and now it's expanded, it's exactly more or less the same proportion?
So when we expand cells or tissues, we are rupturing them. We're disrupting everything, actually, right? We're moving molecules apart from each other. Cell membranes have been expanded, and therefore they're no longer continuous membranes. It is being disrupted from like a life function standpoint. But the information is not being disrupted. The relative organization is being preserved. And that's partly by design. We designed the expansion procedure to be as even as possible. So there's 4 key steps: anchoring, polymerization, softening, and expansion. So the first step is we apply anchors to everything. Those are like little handles. The second step is we form the polymer, which binds those handles. We then could add water, and this is swellable polymer, basically the stuff in baby diapers. So it'll absorb the water, and if it swells and then applies force to the handles, we'd pull the molecules apart. But before we do that, there's a softening step. So we can use enzymes or detergent or heat to soften everything up, and that helps us move them apart. So by design, the process is intended to be as even as possible, but also through lots of validation, right? We can compare to known ground truth, right? Electron microscopy, super resolution microscopy, and now we're almost 10 years into this since the first paper we published on it, and people have validated it in almost every imaginable system. Worms and flies, human tissues. Preprints out there, expanding bone. From a G.B. Chang, a professor at KAIST, who used to be a postdoc in our group. The work that in Silvio Rizzoli's group about expanding protein fragments apart from each other, right? And so far expansion seems to hold up very, very well. And the more it's tested, the more it seems that it's even better than we thought originally. But also expansion can be modified, right? So there's 2 design criteria for a great tool as far as our group's concerned. So first of all, it should be very easy to use, right? Ease of use is important, right? CRISPR wasn't the first genome editor. PCR wasn't the first way of copying DNA. Next-gen sequencing, even by the name, was not the first method of sequencing DNA. But ease of use matters, and in all 3 of those cases, those are much easier to do than their precursors, right? Green fluorescent protein was much easier to use than earlier fluorescent proteins that required small molecule chemicals to be added, and so on and so forth. So expansion, we want it to be very easy to use, and so anybody could adopt it or adapt it if they need to. Also inexpensive. So ideally, a technology would not require hardware that people don't ordinarily have. And again, if you look at a lot of the really great inventions like green fluorescent protein or PCR or next-gen sequencing and so forth, except for next-gen sequencing, I guess, where there are a sequencer does make the process a lot easier, right? But even there, right, the goal is to bring the cost down, and next-gen sequencing, famously, the cost is dropping faster than Moore's law for semiconductors, right? So even though they do require equipment, they still kind of follow the general trend that inexpensive and easy to use really help a technology spread.
Just to put again things in perspective, so for some labs that work on super resolution or they use electron microscopy, one of these tools might cost up to 1-2 million euros or dollars. So it's really restrictive to some other labs where they don't have these resources, right? So therefore, expansion is another wonderful tool that, as you mentioned, it is easy to use and inexpensive, and it gives you incredible resolution if applied in the right way, of course. And it's, mind-blowing. And as I said, this is only the beginning, and I'm very, very curious how this technology will develop in the next 10, 15 years, who knows. And going back now to the connectomics, you mentioned this was one of your kind of wish to understand what is thought, what is memory, and why do you think by knowing the connectomics you would have a better insights to these questions? Why I'm asking this? We know now of course, we don't know if a worm thinks or a fly thinks, but we know the connectomics of a worm and the connectomics of a fly. And still, there are so many things that we do not understand. And the human brain is probably a million times more complex. How is this information going to help you understand thought, for example?
Well, so I think the connectome, the way it's classically been defined, meaning the shapes of the neurons and how they touch, that probably isn't enough, right? You need to know where the molecules are too. Transmitters, receptors, ion channels, right? But I would argue if you have a connectome and you know where the molecules are along the connectome, right? Sometimes we call this the molecularly annotated connectome, or MAC for short. That might be enough to cause a simulation of the brain to work. Because if you know the location of the molecules and you know what they do, thanks to past neuroscience, or maybe assays that you can do in high throughput in cultured cells, let's say, you might be able to then use that combined sets of information to make a model of the brain. And in cases where people have characterized the molecular content, physiology, and connectivity of a neural circuit, maybe most famously Eve Marder and the Krabs somatogastric ganglion, right? So it just has a couple dozen neurons, but they've mapped it. They know its physiology. They know, the ion channels that are found in the cells and so forth. And they can make a model that works, right? So I think that's a great proof of concept that you can make a model with the right information, and it's going to generate, interesting things. But you need the data, right? No data, then I don't think the model would be fully accurate, right? The history of neuroscience, going back to Hodgkin and Huxley, right? Characterizing, the squid axon and being able to model the action potential suggests that if you have the right kind of data, you should be able to make a model.
It's probably hard to compute this and to understand all the locations of particles, proteins, channels. But this is why it is exciting, and it's in front of us to understand. Maybe in our generation or the generation afterwards, we can obtain this information and model the human brain. Who knows? And then understand better ourselves, better the world that we're living in, and of course the laws of nature. So we talked about optogenetics, we talked about expansion microscopy, but you have been involved in so many other discoveries and technologies. What would be your third favorite, if I may ask this question?
Well, we built lots of technologies along the way. When Nir Grossman was a postdoc in our group, we tried to build a non-invasive version of deep brain stimulation using temporal interference. Recently, we've also been trying to answer the question how to image what's going on in the brain. There's so many signals, right? Transmitters, modulators, kinases. Could you image many of them at the same time? And so one area that's very active right now in the group is the idea of multiplex live imaging. How would you image lots of things at the same time in a living cell? And basically, we borrow ideas from how the internet works, how cellular phones work. One way to look at our group is we take classical engineering ideas and implement them through chemistry. So solar panels, that gives us optogenetics if we implement it through chemistry, right? Nanomechanical engineering, that gives us expansion microscopy if we make it through chemistry. And so if you want to image many things at the same time, let's study how the internet and how cell phones work. So I'll just give you one example. So spatial multiplexing, if you have a cell phone and you go from place to place in a city, different cell phone towers will pick up your phone. And that's one way that you can have many people in a city talk at the same time, right? You can have many more people talking than if everybody's fighting for just one antenna, right? And so you can do something similar in a living biological cell, not telecommunication cell. So it says I have a cell here, and I have different places in the cell tell us different signals. This point might tell us calcium. This point might tell us protein kinase A. This point might tell us cyclic AMP, and so forth. I mean, the cell is small, but it's much bigger than a molecule, right? So you could fit many sensors of different kinds in a cell, and if they're different places, you can make all those measurements at the same time while the cell is alive.
So these sensors must be small enough. They must be built in such a way that they do not perturb the physiology of the cell. They need to be able to sense a particular molecule, and they need to be able to report what are they sensing. Is that correct?
Well, fluorescent sensors of molecules have been around for decades, right? Hundreds and hundreds have been made already. Calcium indicators, kinase indicators, small molecule indicators, and so forth. What we're doing is fusing them to self-assembling peptides these are peptides that act sort of like virus proteins, but they're artificial. They're not biologically active. Then the self-assembling peptides will draw the sensors together into puncta, into dots all over the cell. And those dots will then blink out the signal that's found there. So that way each dot can tell you a different story of what's going on. One dot might be telling you calcium, another dot might tell you cyclic AMP, another dot might tell you protein kinase A, and so forth. And all of them are within the cell.
That's
while
incredible.
It's alive.
That is incredible. You can really dissect so many pathways that are happening, or molecules, interactions on spot.
Yeah.
Why would they happen? This is incredible.
Yeah. Another thing we're doing is trying to integrate these imaging tools, perturbative tools, and mapping tools, and to see if we can start to make models of brains. So starting with small brains like worms and fish, where we think it's possible that we could integrate all these tools and be able to do it across the entire brain. So we're already starting to think about how to build pipelines of these tools and integrate them towards whole brain simulations that could be biologically accurate, but that could hopefully generate lots of insights into how the brain works.
Are these peptides somehow expressed? So you manipulate genetically the animal, or you introduce them by any other means to the animal? How do they are implanted, if you wish, in the cell?
This is for the spatial multiplexing?
Yes.
So we can take fluorescent indicators like calcium or cyclic AMP or protein kinase A and fuse the genes to the genes for self-assembling peptides. Then the proteins, just like in optogenetics, will be expressed in the cell, and they then cluster.
Incredible. How far are we, in your opinion, in recording life from a living human brain? I mean, not only living human brain, but also freely behaving. So nowadays we have this kind of MRI machines that record. We have a CT, etc. But this require huge machines that are placed on a certain place, and the individual has to be there, and most of the times not moving. We have, of course, EEG that records electrical activity. But do you think in the future we could have some kind of a portable MRI or other device that can record human brain activity, live? Or can we somehow sense this kind of molecular trafficking in the human brain when we're alive, so to say?
Well, lots of people have built wearable devices for brain imaging using infrared light or EEG or whatnot. So those are out there from many companies. And then implantable devices in prototype form have existed for a while. The Utah array, so-called, has been used to help paralyze patients, to control computers and so forth. So, I think there's a lot of stuff out there already. And I guess one question, of course, is how to make it more accessible and inexpensive and reliable for everyday use. And the other, of course, is can we develop new kinds of things like optogenetics and so forth that can be maybe helpful in different ways, minimizing damage or maximizing the kind of information you can get.
Incredible. I mean, I think we live in a best possible time, right? We have this incredible biotechnological development. AI is kicking in. We have other developments and discoveries in biology that help us understand nature. It's just so exciting to be alive in this kind of era. At least I think so. I wanted to ask you, what do you do outside the lab to keep you sharp? You have so many projects, so many things that are going on around, and you supervise students, and you're involved in a lot of things, and you're doing podcasts. Is there a time for hobbies?
For hobbies?
Yeah.
Yeah. I guess so. I have two children, a 14-year-old son and 12-year-old daughter. So I spend a lot of time doing activities with them, and that's probably one of my main focuses. But I love learning new things, traveling and learning things that are very different from what I might know. So going to, I've gone to conferences and everything from marine biology, natural products from the ocean to, because I'm a big believer in serendipity. The big ideas, the big revolutions in a pre-paradigm science, which I think is what we're in now for the brain, are going to come from very disparate sources, and so you kind of want to engineer serendipity, so to speak, right, and to make it possible to be lucky. So I really try to think a lot about how to teach and help people learn to be lucky, and a lot of our innovations have been driven by serendipity, but the trick is, can you do on purpose what, in the past might have been accidental?
So basically, you try to expose yourself to as much as novelty as is out there, also not related to your field, so that you can trigger some kind of interesting thinking and probably relate to your work or teach somebody some meaningful lesson. This is basically what you said. Am I right?
Yeah, but not just novelty. I mean, it can also be very old things too, right? So, the physics of the responsive polymers for expansion microscopy. The key paper was a 1980 paper. The opsins for optogenetics. The first was discovered in a 1971 paper. So, sometimes novelty means looking at old things.
Incredible. There is a question that I'd like to ask you also about a certain thing. So imagine you have resources to run an institute or a company for 10 to 15 years, let's say. You have the unlimited resources at that time and everything is governed by your desire and wish. How would this institute/company would look like?
Well, first of all, I really do think that if you want to engineer serendipity, you have to really be distributed. So I probably would want to have a core, let's say in a place like Boston, where there's a constant stream of people arriving and departing at all times, but also the ability to dynamically reconfigure. So could we distribute resources to collaborators all over the world? People often talk about, could you copy Bell Labs, right? So Bell Labs famously did the transistor. I think they did the precursor to the laser. They discovered the background radiation that was the first real, I guess, indicator of the Big Bang. And people often say, oh, how do we copy that? So I think the trick is when you create a system, it has to be appropriate for the problem at hand. And I feel like every problem has a natural home. Problems of the 21st century, I feel, are made of so many more parts and interactions than, let's say, physics or other sciences that matured in large part in the 20th century, let's say. So, so I think we do need to have a distributed component that can be rapidly reconfigured.
So stream of people that are highly driven, self-driven, collaborative environment, and of course, this kind of innovative ideas you would basically support. Interesting.
Well, if you look at the history of biology, right, you can see where the innovations came from. And they often came from very different sources. They came from, collaborations. They came from different people, from different backgrounds. And so if one wants to systematize that, then I think that could be done. But one would have to, take a look at what historically worked in biology and then see if we could scale that up.
How different backgrounds have the people in your lab? Is there anything that is completely different? Is there a person that has like a background in, let's say, photography, or although photography is highly related to what you're doing, imaging, something that is completely unrelated but works in your lab?
Yeah, we had a PhD student who was a professional photographer before joining my group. We've had people join the group with no bachelor's degree. We have two people who dropped out of college, one of whom just finished the PhD in my group, and the other is heading in the home stretch. So
that's very interesting. So people need to be just smart and encouraging and reach out and hey, maybe you end up in Ed Boyden's lab. Who knows?
Yeah. Well, what matters are three things, right? A quest for the ground truth, the ability to change directions, because that's what we do, and then the willingness to go in new directions.
To go and work hard, actually. I heard somewhere online in the online field, some of your interviews in the past, you mentioned that you started the very first experiment or one of the experiments in optogenetics very, very late at 1 a.m. in the morning. Is that true?
Yeah, in 2004. Yeah.
Incredible. So guys, you have to work hard and work also late or early in the morning. Yeah. There's another unsolved mysteries around the human brain, and this is consciousness. What is your take on that one?
Yeah, well, that's kind of why I got into neuroscience, right? To understand philosophy, and that's one of the core philosophical questions of the brain. I think the tricky part about consciousness, right, is that we cannot measure it. You don't even know if I'm conscious, right? Maybe I'm just a very realistic robot with ChatGPT version 6 and something like that. But I think that's one of the big questions, right? That's one reason why I'm excited about simulations, right? Because maybe you can look in the simulation, see patterns of activity that don't correlate with any known behavior or outwardly facing process. And maybe those could be candidate signals to look for and to see whether they could be associated with something like subjectivity.
Do you believe it's more like, there are these hypotheses, dualism or theories, if you wish, materialism. People think that this emerges. It's kind of an emergent property of the brain activity. You have some other theories like the integrated information theory from Tononi. You have a very interesting take on consciousness from Donald Hoffman, who claims that consciousness is the first, and out of it emerged the physical world, which is also like completely inverted version of the physicalism, if you wish. Do you think the truth is somewhere, probably somewhere in between? But what do you think is the actual fundamental truth of consciousness?
I don't think I'm ready to have thoughts about that. I prefer to be driven by the data. So I think Sidney Brenner once said, first you have tools, then you have data, then you have theory, in that order. I think that if you look at the history of science, that's actually quite often true. I mean, even in physics, right? The most theoretical of the sciences. People look at Newton's laws, for example. But before Newton's laws, right, you have people like Kepler and others trying to explain the movement of the planets and coming up with proximal theories based upon those data. And before that, you have centuries of astronomers looking at the sky and so forth. So if even the most theoretical of the sciences needs to have a firm foundation of data to build from, why do we think biology is any different?
That's a great question. Great answer to this question. I love it. Professor Boyden, we are approaching the end of our first part of the podcast, and I'd like to thank you once again for your time and for being here with us today.